welcome to the Data Science Seminar from the University of Washington eScience Institute. Joining us today from remote is Dr. Komp, who recently received his PhD in chemical engineering data science at the University of Washington, working with Dr. David Beck. He was awarded a data science fellowship from the Clean Energy Institute and performed work as a machine learning engineer in pharma. He has worked on several topics at the intersection of deep learning and chemical sciences, including molecular properties, chemical reaction rates, and protein thermal stability. Ivan is a strong advocate for the use of data science to help develop a more sustainable, climate resilient, and friendly society. And I like this message in his bio because he is encouraging everyone who works in a compute-intensive environment to track their computations' carbon emissions. Thank you, Ivan. You can take it away. Welcome, Ivan.
Awesome. What's that? Awesome. Thanks for the introduction. It's a pleasure to be here. Nicoletta introduced sort of three different things that I've worked on at the intersection of deep learning and the chemical sciences. And I know it's typical for a sort of seminar like this for somebody to go over, 2 to 3 stories that they've worked on over their professional career, but I'm actually going to focus on the most recent one that I've been working on for about the last year and a half here, because I think that it's the most exciting. So with that little change of pace, hopefully we can get into a little bit more nitty-gritty details than you're sort of accustomed to for a seminar like this, and hopefully it's a nice stimulating talk. Bummed I can't see the faces in the room. I'm sure some of you have already seen some of this content already, so hopefully there'll be a nugget of something new in here. So I want to get everybody on the same page about proteins and why proteins. So 20 different amino acids, which you can think of as sort of like little Lego bricks, little small molecule bricks that can be chained together, achieve a huge diversity of different functions in your cells, as well as the cells of all the other organisms on this planet. And the proteins are created by translation at the ribosome, where a very specific sequence of these amino acids are chained together. And that sequence is determined by a protein encoding gene in your genome. Now, once translation has occurred and we have a amino acid sequence, which we call the primary structure of the protein, in its native environment, it will what's called fold up. So at a sort of local scale, you have secondary structure elements like helices and sheets. And then at a larger scale, what we call the tertiary structure is essentially the three-dimensional shape of the single protein in its environment. And this three-dimensional structure allows for this diverse set of functions that I mentioned. For example, we could observe a binding pocket for a particular transition state to enable a reaction, or bind to a small molecule or even another protein. So not only do these proteins achieve a huge degree of functions within your cells, but also as leveraged by us sort of as humans. So we can consider, for example, industrial enzymes. This was a $6.6 billion market in 2021. And I would say all. I'm not sure if I can make the statement all, but all that I know of, all the applications that I know of, like to have these industrial enzymes operate at an elevated temperature. So that right there is a cellulase, and it's not just any cellulase. It's actually one that came from an organism called Pyrococcus furiosus. And this enzyme remains active at 102 degrees Celsius. So that means it can continue to break down the cellulose polymer at a boiling point. We can also think about therapeutics, which is seeing an increased use of peptides and proteins for, vaccines, diagnostic tools, the list goes on. And for example, Gardasil, which is the HPV vaccine and contains peptide components, a really resource-intensive and carbon-intensive cold chain is required to sort of keep these components cool from point A to point B as they're shipped around the country. So that is the spike protein of the SARS virus, but it's actually not the native one. It's the one that's been engineered by us humans in order to be stable for transport up to 60 degrees Celsius, such that, you can ship this thing around without having to worry about a cold chain, such that people can continue to research vaccines and other tools on the protein. Unfortunately, having proteins that are actually stable at high temperatures is not ubiquitous. So as dictated by Gibbs free energy equation here, if ever the unfolded state of the protein or some non-native folded state of the protein becomes less in Gibbs free energy, then the equilibrium will shift away from the folded functional form that you want, and you'll no longer have the protein doing what you care about. For example, you can think about the melting temperature. This is the temperature at which this equilibrium shift occurs. And if you go out there and look at all of the melting temperatures that we've measured for all the protein, well, not all the proteins, but if you go find all of the melting temperature measurements that we've made, which is actually not that many of them because it's quite expensive, you get an average of about 60 degrees Celsius. So though it's not ubiquitous, we know that it's possible because there are organisms called thermophiles that thrive at extreme temperatures. For example, Pyridictium abyssei actually lives at 105 degrees Celsius, which is kind of crazy. And the question is, how do these thermophiles adapt their proteins to remain functional at these elevated temperatures? And is it possible for us to learn from the proteins that they have in order to help us engineer our own? So when I say mesophile, I mean an organism that grows at ambient temperatures. And when I say thermophile, I mean one that grows at, say, above 60 degrees Celsius. Now, the method that I'm going to be talking about today is to use neural machine translation, which you may have heard of in natural language processing context, to try to fit this niche. So to be very explicit, our engineering goal is we want a method to rapidly develop high temperature variants of some target protein, and we want these methods to be fast. That means we don't have to put much time or effort into them. We want them to be precise such that the time and the effort that we do put into it is actually going towards a protein that is going to be useful and not some off-target protein. And we also want it to be reliable. So that means that when you come to me with a protein, we can reliably get to some engineered variant that is more thermally stable. Now, the way that we're approaching this is, again, with neural machine translation, which in the sort of canonical example occurs in natural language processing, where you would have synonymous phrases in one human written language like English going into the translator. And once trained, that translator will produce for you synonymous phrases in some other target language like French. The analogy here, and this is not necessarily a new analogy, is that proteins and amino acids are kind of like words in sentences. But what is novel is to treat it as a neural machine translation task, wherein, furthering this analogy, we can imagine that mesophiles and thermophiles have a sort of different dialect of amino acids that they use to produce their proteins. So before I actually talk about the method, I wanted to sort of go through, at least at a very high level, the methods that already exist to specifically address this engineering goal. People have been trying to meet this goal for decades, and we've made lots of progress. And I also want to talk about how deep learning has already helped some of these processes recently, and what hole there is that I believe this work helps to fill. So if you come to me with a wild type or some target protein that you want to create or engineer to be more stable, the methods that you can use, I would argue, sort of bend into these sort of two buckets here. The first is methods wherein you're introducing lots of variation to the protein, or maybe you have many different variants that you want to test. And then you actually go in at the bench and, synthesize and measure them to see how well they do at the thing that you care about. For example, are they stable at a higher temperature? And the ones for which that is true, you would then continue on to increase the amount of changes that you're making to it over time. Now, if you do a process like this with random mutations upon the sequence, that's what you've probably heard of as called directed evolution. The other bucket here at a really high level is, instead of basically making changes to the protein and seeing which ones work, is to try to use things that we do know about proteins, like first principles, how atoms interact with each other, maybe fitted energy functions, in order to directly try to engineer the protein to be more stable. So that is like find positions within the protein that we think need to change in order to increase its stability, introduce mutations at those positions that we think will be helpful, and then see if it does. Now, this, of course, is a sort of faster process because we can do it in the computer and it doesn't take a huge amount of lab effort, but it's not nearly as reliable. We're not guaranteed to get to a particular temperature in terms of our stability, and we're not even guaranteed to actually increase the stability at all. And that's just simply because, well, proteins are really complex and we don't know everything about them. So with these two sort of broad buckets in mind, I want to dive into just a little bit more detail into the right bucket. And that is that there are methods that you can use to produce variants that are non-random. And this sort of minimizes, or doesn't minimize, but it reduces the amount of time that you're spending testing variants that are detrimental. So when you're randomly mutating the protein, over 98% of the time, a lot of the time, those mutations are actually going to be not helpful. And so we want to try to avoid that. And one way that we can try to do that is to introduce variation using homology. So in this sort of setup, we would have the target protein that we're trying to engineer, and we would scan it against a sort of massive data set of all the proteins that we know about. And if we're lucky, we get a large number of homologs, which you can think of as sequences that are likely related to the target protein sequence evolutionarily. And then with those homologs, we could, for example, just screen those homologs. Some of them may be actually better at the thing that we're trying to do than our target protein sequence. We could also take those homologs and use the changes that are occurring on those homologs compared to our original sequence and use those as our sort of introduction of variation into the protein sequence that is non-random. And this reduces the amount of time that we're testing proteins that are basically just detrimental, but there are still many variants to test. On the other hand, we could try to, for example, align these homologs into a multiple sequence alignment and then use that to create a consensus model of the protein. And this is, you can think of this as essentially what are the positions and the amino acids at those positions that are conserved across all of these different homologs, in which case we definitely need to keep those ones. But when we create a protein this way, it's not guaranteed to be actually more stable than the target protein. And further, both of these methods that I sort of briefly mentioned here require that you can actually find those homologs in the first place. So if you're in a situation where your target protein is not one where you can find a bunch of homologs, then you're out of luck and you have to resort to these sort of random mutations as you would with the very basic form of directed evolution. And this is part of the niche that the neural machine translator aims to fix. That is that we don't actually have to go find a bunch of homologs in order to use it. So keep your eye out for that sort of truth about the neural machine translator moving forward. Now, deep learning applications have helped with these processes, but deep learning applications require lots of data. When it comes to proteins, we know about hundreds of millions of protein sequences, billions if you consider metagenomic data sets where we don't actually know their source organism. We've also measured an extreme number, well, extreme's not a good word here, but we've measured hundreds of thousands of three-dimensional structures of proteins. And if you consider high accuracy predictions, then of course this is an even larger number. So it's safe to say that we're definitely in the sort of big data era when it comes to proteins themselves. But what about the thing that we care about, which is high temperature stability? Well, if you have a protein that's expressed by a particular organism, you could try to measure its melting temperature with something like differential scanning calorimetry. Again, this is a very expensive process, and the result is that the largest data sets out there are about 48,000. And even within that data set, it's not a particular diverse set of proteins. So we're not really there when it comes to thermal stability targets for deep learning applications. But we could consider taking the organism that expresses that particular protein and measuring at what rate it grows as a function of temperature to find its optimal growth temperature. And we know that while the proteins that are expressed by that organism occupy a distribution of different melting temperatures, they have to be greater than that optimal growth temperature. Otherwise, those proteins would not be functional where the organism is trying to grow. And so this is clearly a much more noisy metric for thermal stability than melting temperature, but increases the amount of data that we have significantly. So these data have been used to help accelerate these processes. For example, when it comes to a directed evolution campaign, you can actually train predictors of the thing that you care about, whether that be some activity or stability metric, as the directed evolution campaign moves forward. And so that means that after the first few rounds of evolution, you actually have a predictor in place that can help you filter out many of the variants that are likely to be detrimental before you even actually have to spend the time to test them at the bench. We've also, in the last about 2-3 years, gotten to the point where we can create generative models that actually introduce protein variation into our sequences directly instead of having to go acquire it from homologs. However, the methods that exist don't actually specifically target high temperature stability. They're just generating it from learned variation. And hopefully that makes more sense moving forward. So this has definitely accelerated the process of running these style of engineering campaigns. On the other side of things, we've had for a long time machine learning models that help us evaluate a sort of score or an energy function for a particular sequence in a particular pose in a particular three-dimensional format. And that helps us iterate through the process of designing a primary and tertiary structure to meet something that we care about. Whether that actually plays out at the bench, sometimes it works, sometimes it doesn't. We've even gotten to the point where there are models out there. You've probably heard of, AlphaFold, or maybe you've also heard of something like protein MPNN, where you can have a single model translate directly from sequence to structure and vice versa. And so this is definitely speeding up the process of trying to directly engineer a protein. Unfortunately, even though deep learning has helped, none of the methods that are out there specifically target high temperature stability. And for the case of the directed evolution style approaches, they usually require homologs in order to sort of speed up this evolution above random. So that's where the neural machine translator comes in. While there's lots of data for deep learning, they are not in the format necessary to train a neural machine translator. So I'm going to start off by talking about how we produce such a data set by joining experimental growth temperature data with genomic data, as well as what is initially an intractable space of proteins to search for, how we narrow that down and actually find pairs of proteins occurring across mesophilic and thermophilic organisms in order to try to train a neural machine translator. So first here, before actually training the translator, it's worth talking about trying to determine the resolution and growth temperature that's necessary to actually differentiate these protein sequences. And then I'll talk briefly about training the translator itself. And then, of course, hopefully spend 10 to 15 minutes talking about how the translator can be used to help us engineer some protein variants. So I'm going to skip the first part here because this is literally just a data joining exercise. It's like primary, foreign, key. That's oversimplifying it a little bit, but suffice it to say that it's not too interesting to talk about. I want to start with what is initially an intractable protein space. And so what I mean by that is that after collecting all this data and doing the data joining, we essentially have a huge data set of proteins whose source organism we know, and that organism's growth temperature we also know. So we have a big bucket of thermophilic proteins and a big bucket of mesophilic proteins. And we want to try to determine across those two buckets which ones are homologous, which ones are actually related to each other. Now, how do we do that? Well, if we want to determine if two proteins are doing the same function, we can think about their homology, which is on the right track, but not equal to function. So for example, we could align two sequences and determine how similar they are to each other. And we can also align many sequences, which gives us a little bit more sensitivity. We also might think about comparing the three-dimensional structure of the proteins. If they're significantly different, then they're probably not doing the same thing. And again, we can align two or multiple three-dimensional structures. And we're essentially asking the questions, do the residues in these two to two plus structures overlap with each other? Do they occupy the same three-dimensional fold? The gold standard, of course, would be to study in both of these ways a particular pair of proteins. But suffice it to say that we have a huge search space to search over, and there's no way that we could possibly run a structural comparison over all of them. Even if we consider the fact that we can start predicting three-dimensional structures these days, they're quite expensive to do. And even on the homology side, we ran a test of all of the mesophilic proteins versus all of the thermophilic proteins and found that it would take about 5,000 hours using 48 high-performance computing nodes on our own HIAC cluster. So I hope you acknowledge with me that that's an unacceptable cost. So we had to think about how to determine which parts of that very large pairwise protein space to actually focus our search. And what we landed on is, instead of starting with pairs of proteins, we looked for pairs of organisms. So every organism has a 16S rRNA sequence. This is a sequence that has been used previously to infer phylogenetic relationships, and it's very, very slow to evolve. So what we did was we aligned the sequences among mesophiles versus thermophiles, and if the 16S rRNA was highly similar to each other, then we would say that those two organisms are related to each other and probably worthwhile to search for similar proteins among that pair of organisms. And it's only for those pairs that we actually ran alignments of proteins in order to find protein pairs. The result is 69 million protein pairs stemming from 150,000 pairs of organisms. And some things I want to point out about the process of creating this data set is that the entire thing is tracked in what's called data version control. This goes one step further than your standard version control of code, and that you can actually see how the data itself changes as you write the code, as well as how you change, or excuse me, how the data changes as you change parameters. It also means that you or anybody else could go into our code base and execute the entire process of creating this data set from the data joining to the alignments to some downstream validation steps that I do not talk about here with a single command line command. Also, we tracked the estimated amount of carbon it took to run these alignments and found it to be about 7.2 kilograms of CO2, which is a non-insignificant 0.02% of a United States household's yearly carbon budget. Keep in mind that a United States household is far and away the most emissions of any country in the world. So this data set is both deep and wide. On the left, you're looking at a breakdown of the different organisms that are contributing to the data set. So archaea and bacteria are included, and you can see a breakdown of phylum down to order on these sort of outer rings here. There are many different orders of organisms that are contributing. And then on the middle, every single one of these green lines that you see is actually a connection between a mesophile and a thermophile. So this is a pair of organisms that we found to be related to each other. And it's for those organisms that we search for protein pairs. On the right, you can see a two-dimensional mapping of the proteins that are contributing to protein pairs. So in blue, you can think of this as basically the sum of human knowledge of proteins for which we have a high degree of confidence on the three-dimensional structure. In red, you can see the previous largest data set of protein pairs, which is far too few to actually think about training a neural machine translator. And then in yellow, you can see our data, which you'll note that there's some holes compared to the blue, but it's covering a significant portion of this space, and it's also very redundant. So there's lots of variety within a particular niche. So this gives us a data set of more than 4 million. I said 69 million previously. We filtered it down to the highest quality pairs, which is about 4 million, versus the previous largest data, 1.6 thousand. So this is a number that we can actually think about training something like a neural machine translator, which is transformer based. Before talking about the translator itself, I want to talk to you about trying to basically make sure that the growth temperature labels that we acquired were good ones. And so to do this, we split the data set up into proteins that occur in organisms below 30 degrees Celsius and those that occur in organisms above 60 degrees Celsius. And on a held out test set, which is actually split by the organism itself, such that the model could not memorize organism level patterns, we find that we can predict with a very high accuracy whether or not the protein came from a mesophile or a thermophile based solely on the protein sequence with an F1 score of 0.91. The confusion matrix here on the right, we want the bulk of the probability mass to be along the diagonal, and indeed it is. Okay, so we know that this data has signal. Let's actually train a neural machine translator. What does it do? Well, a neural machine translator is going to take in pairs of text, in this case, a pair of a mesophilic homolog and a thermophilic homolog to train it. The mesophilic homolog is given to an encoder, which essentially takes its distilled knowledge of protein sequences and represents the mesophilic input to the decoder. And the decoder's job is to predict the next amino acid, and it does so. And then that amino acid goes back into the input of the decoder, and it repeats this process. It predicts the next amino acid. And this process repeats until the decoder itself decides to stop, after which point we hope that the produced sequence is similar to the ground truth thermophilic homolog, but not perfect. We don't want it, maybe counterintuitively, to be the exact thermophilic output sequence because our situation looks a little bit more like this. Evolution creates a lot of variation, and we have these large groups of mesophilic homologs compared to large groups of thermophilic homologs. So if the decoder perfectly reproduced a particular thermophilic sequence given a input mesophilic sequence, it would do poorer on the other ones. What we actually want it to do is to sort of learn the average of thermophilicity over evolution. Okay, so to try to measure this and give us something to target, you can think about the cross entropy from natural thermophilic diversity, and that number is 0.94. So just to ground your knowledge in what this metric means, cross entropy of 0.0 means that in every position, the amino acid is exactly predicted correctly with 100% confidence. Whereas if you take the thermophilic sequences in our test set and you search against natural diversity, and then you create multiple sequence alignments on them and measure the diversity of amino acids, you get a cross entropy of 0.94. So that's the number that we want to achieve, except again, that thing that I mentioned previously, without having to input many homologs. We only want to input a single sequence, which is what, that's what our model is attempting to do, whereas searching over natural diversity like this required to search over homologs. Okay, so we split the data set by sequence identity such that there is no sequence in the test data set that is highly identical to in the training data set to hopefully give us a good reproduction of how well the model's doing on unseen data. And before I actually talk about the scoring of the model and how well it's doing, I just want to mention that if you've worked with transformers before, you know this, they're quite expensive to train. So training this model took, after a few different training runs, summed together about 102 kilograms of CO2, which is 0.2% of a United States household. Okay, so let's actually use the translator. Well, I told you that we're shooting for a cross entropy of 0.94, and we achieved that with 0.9. Again, I'll reiterate, the model takes only a single input mesophilic sequence, whereas this second number, 0.94, was achieved by searching over lots of homologs for every single mesophilic input. Okay, now we want to go further than the sort of canonical natural language processing scores like a BLEU score. That's not very applicable here. So instead what we did was we took the sequences that were produced by the model and compared them to the ground truth thermophilic targets with a Smith and Waterman alignment and found that the sequences produced by the model are 43% identical in terms of their amino acids to the thermophilic ones. And this is a pretty good place to be because too much lower than this, and we would have to get concerned that on average, the sequences produced by the model are probably in different functional niches, whereas too much higher than this, and we might worry that the model is sort of memorizing sequences. We also wanted to compare the three-dimensional structure to the best of our ability. There's no way we could possibly measure the three-dimensional structure in any sort of throughput for this data set size, but we can predict them with these sort of recent state-of-the-art structural prediction tools. And once done so with ESM2, we basically determined the secondary structure of the proteins using DSSP, and we determined the Jensen-Shannon divergence between the proportions of secondary structure used in the thermophilic proteins and each corresponding one produced by the model is 0.01. That's indicating to us that the secondary structures produced by the model are very similar for every example in the data set to the thermophilic ground truth. And then if you just compare the full structure with a three-dimensional structure alignment, you get a p-value of 0.1 using this software called FatCat, which is telling us that the three-dimensional structures are similar to the ground truth thermophilic target as well, at least according to ESM prediction. Now, people have been we've been searching for decades for the things that actually make thermophilic proteins thermophilic. What is it that makes them stable at high temperatures? And suffice it to say that there has been a lot of different work. A lot of that work is contradictory to other work. There are many things that have come out that, works in one case and not another. One thing that has been sort of accepted by the literature, though, is that thermophiles just simply tend to use amino acids at a different propensity than do mesophiles. So in black here, you can see the shift in propensity of each of these amino acids on the x-axis from mesophilic to thermophilic. And then in orange and purple, you can see our data set as well as the proteins produced by the model. They are in the same direction and in most cases the same magnitude as the literature observed variance for all of the statistically significant propensity shifts, which are highlighted in purple here. One of the other things that has been noted about thermophilic proteins is that thermophiles tend to use disulfide bonds to a greater degree than do mesophiles. And so a disulfide bond forms or can form when two cysteines are near each other in space. Whereas when a cysteine is not near another cysteine, it does not form a disulfide bond. These sort of lone cysteines can have other amino acids substituted in for them relatively analogously without hugely affecting the shape or the three-dimensional structure of the protein. Whereas when you substitute out a cysteine that's forming a disulfide bond, you're much more likely to severely change the way that the protein is working. So we want to see that the model can actually recognize this. So we took in our test data set all of the cysteines And we found which ones we hypothesized were forming disulfide bonds, essentially by a heuristic distance. And we binned all of those cysteines. And we wanted to see that the model recognized that cysteines that are forming disulfide bonds are extremely important. And indeed, that's what we see. So for the non-bonding case of cysteine, the model is a little bit more likely than random on average to place a cysteine in that location, whereas for cysteines that form disulfide bonds, the bulk of the probability mass here is greater than random. That means that, keep in mind, this is the log scale down here. That means that the model recognizes that cysteines must occur in these positions where disulfide bonds are likely being formed in order to for the protein to achieve its structure. Of course, the thing that we actually care about is not, how well the model is identifying these, things that we found in literature tend to happen in thermophiles, but whether or not the model can help us actually produce thermally stabilized proteins. So to do this, we used a very recent sort of state-of-the-art in silico estimator of stability, which is actually a really cool method that I want to go over briefly because we use it to evaluate some of our designs. That is to take the input sequence, predict a three-dimensional structure using AlphaFold many times, basically creating an ensemble of structures, given that this is a stochastic process. And then for each of those structures, minimizing the structure in Rosetta's energy function, which outputs for you a score, and taking the mean value of that score actually results in something that is extremely qualitative to experimental thermal stability targets like melting temperature for a number of different protein families. So this is quite promising, and we use it to sort of help evaluate some of our proteins moving forward. So we took this thermal stability estimator and we ran it on the mesophilic inputs, the actual ground truth thermophilic homologs, as well as the ones that are produced by, the proteins that are produced by the model in our data set. That gives us 3 scores. And we want to see that the thermophilic and the model generated scores are better than the mesophilic ones, i.e. they're more stable, at least according to this estimator. And indeed, that's what we find. So the thermophilic homologs on average tend to be more stable. And it seems as if the generated protein sequences by the model are actually even slightly more stable according to this estimator. We hypothesize that this is because a single thermophilic sequence is sort of one trajectory in evolution, whereas the model is learning from many different homologs. It's learning over variation in evolutionary space, and so it can sort of focus on the average, that is, the things that are actually making it more thermophilic as opposed to the sort of things that have changed randomly. Okay, so let's actually use it to engineer a particular protein. This is Engrailed Homeodomain. It's a 3-helix bundle transcription factor, which means it binds to DNA. And this has been extensively studied in the past. So people have run all sorts of simulations on this protein. They've previously engineered them to be more stable. And if you run the in silico estimator that I talked about on the last 2 slides on the wild type protein sequence and compare it to one that we found in literature, which had been previously engineered using consensus modeling, you can see that the in silico estimator resolves the fact that it thinks that the consensus engineered variant is more stable than the wild type with statistical significance. If you give the engrailed homeodomain wild type to NOMELT as if it were a mesophilic sequence, keep in mind that this is a eukaryotic protein for which the model never saw any eukaryotic proteins. It outputs what it thinks is a sort of thermophilic variant of that protein. And if we run the estimator on that protein, we get about the same score as the wild type, which is sort of discouraging at first, but you have to remember that the model is introducing a number of mutations on the sequence, in this case, 14. And if you were to randomly introduce 14 mutations into this protein sequence, it's significantly destabilizing. So the fact that we're introducing 14 mutations and it's not destabilizing the protein is actually promising, and it sort of points towards that thing that I mentioned at the start of this presentation. We want a way to introduce variation that is better than random. So if we treat these 14 mutations as sort of, variants to test and make a combinatorial library of those 14 mutations resulting in 16,000 different variants, and we search over only 100 out of 16,000, again, 100 out of 16,000 possible variants using that in silico estimator that I talked about in the previous slide, as well as a sort of optimization algorithm. After only 100, we find that we can achieve a stability score that is statistically no different from that one that we found within the literature. And if you repeat this process for the 14 random mutations, you can't do better than the wild type score with statistical significance. So again, better than random introduction of protein variation, at least according to this in silico estimator. So I want to talk to you about some molecular dynamics simulations that we ran on the protein variants that we are presenting on here for Engrailed Homeodomain. So in molecular dynamics, we are inputting essentially a three-dimensional position of the protein with basically where are the atoms in three-dimensional space. Then we set up a sort of simulation such as what is the temperature of the simulation, as well as a number of other parameters. And then essentially, over a minute amount of time, we allow the interatomic forces of the atoms within the protein to exert themselves on each other. We can calculate a force, we can add those forces, or we can determine an acceleration, add the acceleration over a period of time to any existing velocity, and we can watch the atoms move over that very small period of time. And then we can repeat that process, essentially giving us a sort of movie of the dynamics of the protein according to, whatever fitted force field we're using. Now, at any point, we can compare the three-dimensional structure of the protein to the starting structure and compute what's called the root mean squared deviation, where a higher number is indicating a larger difference of the structure compared to the starting structure. And so on the next slide, I'm going to show you some pictures that look like this. In full opacity, you're essentially seeing the starting structure, and then each of the translucent structures in the background are the resulting structure after a long simulation of the protein. And we did this in sort of replicates of 5. So here you can see this is a low RMSD ensemble of simulations because all of the structures at the end of the simulation are very similar to the starting structure, whereas the RMSD is much higher in this ensemble of simulations, where you can see that the helices are significantly changing their pose compared to each other. Okay, so let's actually look at the results. Christian Phillips ran ensembles of 5 simulations for a range of different temperatures for the wild-type Engrailed Homeodomain, as well as one that has been previously engineered that we found in literature to a greater than 98 degrees Celsius melting temperature. Now, the further that you move away from y equals 0 here is essentially further away from the behavior of the protein in terms of its RMSD at 298 Kelvin. So what you'll notice is that the previously engineered variant, both the magnitude and the width of the RMSD values that we observed remains relatively close to the RMSD values at 298 Kelvin. Whereas for the wild type, at the protein's melting temperature, both the magnitude and the width of the distribution of RMSD values that we noticed jumps up significantly. You can actually see that here, wherein for the wild type, if you're looking at the three-dimensional structure at the end of the simulations, the helices are significantly sort of detached from the starting structure. Now, we repeated this process for the protein that we engineered on the previous 2 slides ago that I was talking about. And what we find is that at the wild type melting temperature, we have ending structures that are very similar to the starting structure, so very low RMSD. And it takes about 10 degrees Celsius more before we see that jump in both magnitude and width of RMSD values. So it's safe to say that this variant that we've produced fluctuates less according to molecular dynamics, but 2 things I want to point out. First, over here on the right is that molecular dynamics is great. It helps us sort of study some things, but it's not experiment. So experimental validation is required, and we need to actually find some experimental data, which I'll show you on the next slide. And then the second thing I want to point out is that MD simulations are quite expensive. So about 304 kilograms of CO2 went into running these simulations. Okay, so let's actually get some experimental data in here while we're trying to develop the resources necessary to actually express some of the variants created by this model. We went and found existing melting temperature and other thermal stability target data out there. So here is what you're seeing here is an enzyme called LOVD. It catalyzes a inert intermediate into levastatin, which, if I remember correctly, is a high blood pressure drug. And oh, I'm missing a citation here. Sorry, guys. If you're curious to find this data, I'm happy to send it after the presentation. The authors actually looked at LOVD here, and what they did was they ran directed evolution on it, and they accumulated a number of mutations upon the wild type sequence. And this one up here that I'm pointing at, hopefully you can see my mouse here, is 15 degrees Celsius higher in melting temperature than the wild type, and it accumulated 29 mutations upon the wild type sequence. Now let's talk about the y-axis here. This is essentially the output of the model in terms of the probability of the amino acids at those specific positions in those variants. So this is not what the model was trained to do. Again, the model was trained to translate between mesophilic to thermophilic protein sequences. But what we did was we took these existing variants and evaluated how likely a particular amino acid would be to occur in that sequence to make it look like a thermophilic sequence. And what you find is that there's a extremely tight correlation between the output of the model and actual experimental melting temperatures for LUVD, which is kind of crazy. Again, this model was never given melting temperatures ever. It wasn't even given a thermal stability target to predict. It was trained as a translator. Unfortunately, it doesn't work every time. So lipase A is a enzyme that breaks down lipids, breaks down fatty acids, or it aids with that process. And it can sort of qualitatively evaluate the highest melting temperature variance, of which the highest accumulates 12 mutations upon the wild type sequence, but it severely overestimates the wild type score. We hypothesize that that is because it's already quite high. Lipase A has also actually been the focus of a deep mutational scan. So unlike the data on the left, here, a deep mutational scan is a bunch of protein variants for which there's only a single amino acid difference compared to the wild type. So all of these variants are very, very similar to the wild type. They're not actually accumulating mutations. But there's an existing data set out there of catalytic T50, which is the temperature that you reach half activation for that enzyme for lipase A. And Hamoud here ran the model predictions on this benchmark data set and found a Spearman correlation coefficient of 0.32, which is actually state of the art. So ESM2, which is a protein language model, it's a BERT style language model. It's not trained as a translator like ours was trained on more than 65 million protein sequences, and it achieves the same score as we did with only 4 million protein pairs. So this is sort of indicating to us that approaching the task as a mesophile to thermophilic translation task, as opposed to just trying to learn all of the variation across nature without any labels, is sort of advantageous. So there are models that can do better, but they require known variation as input. So remember, one of the themes of this talk is that our method does not require that you know a bunch of homologs of your target protein in order to use it. So there are models that sort of do that, right? Our model, as well as ESM, takes just a single input sequence, gives you a score. There are models that first you search for homology, and as long as you have a sort of deep enough multiple sequence alignment, you found enough homologs, those models can score even better. But again, they require homologs as input. Okay, so I hope I've convinced you at this point that there is a niche left to be filled within using deep learning to help us engineer high temperature stability proteins, and that is ones that focus on high temperature stability and do not require many homologs as input, and that our neural machine translation model is sort of on that path. So I would love to acknowledge my amazing advisor, Dave Beck, who I do believe is in the room, as well as all my collaborators on this project, Hamoud, Christian, Logan, Chow, and Ryan, as well as Marlo. They all contributed heavily to this project. And then I also want to point out that we estimate that about 500 kilograms of excess compute carbon was used to actually conduct this work. All right, any questions? Wow, I hope I was connected to the seminar this whole time.
Thank you, Evan. That was glorious, I'd say. Questions for Evan? And if you do have them from up there, please come down to the middle of the room so Evan can hear you.
I'll get started.
Okay.
Evan, how could you incorporate structural information into?
How could you?
Yeah, I got to write my next grant.
Well, there's a number of different deep learning methods out there that can actually take as input three-dimensional structure. So many of them kind of look like transformers, like I've been talking about, except they have to incorporate some way to basically capture invariance or equivariance of the structure. I suppose what I'm telling you is that you could basically change, you could put in parallel and input a language modeling input like we have in the encoder, as well as a, message passing neural network or a three-dimensional transformer right next to it. And you could give both of those pieces of information to the model in order to have the decoder produce for you a thermophilic variant. I think there's a lot of ways you could sort of approach that, though. I think suffice it to say that we have machine learning models that can operate on three-dimensional structure. I think the problem is the source of the data. So you would, in order to really operate on the scales that we would need, you'd have to make some people argue, perfectly fine assumption that AlphaFold predictions are ground truth. But you could go that route.
Hi, I was wondering if you know of any existing studies on assessing the function or efficacy of meso versus thermophilic proteins at different temperatures, like including the ones more for the mesophilic.
If I'm understanding the question correctly, how do you determine whether or not a protein is, achieving some function at a particular temperature and how it changes as a function of temperature? Did I get that right?
Yeah, I guess I was wondering if you want to be able to translate these, but do you want the proteins to still be functional at lower temperatures, or are they only functional at the higher temperatures?
I see. That's an absolutely fantastic question. So one of the things that I sort of glanced over a little bit here is that just because a protein is, more stable, maybe it's, more rigid and it doesn't melt quite as readily at some high temperature, that doesn't mean that the protein is actually active or to what degree it's active at a higher temperature. And the same can be said for a lower temperature. So this is a sort of, this is a very tightly bound problem. And that's exactly why I am sort of presenting this method as a way to introduce variation into existing sort of pipelines wherein you would actually go evaluate those things. So you wouldn't want to take the output of this model and assume that it's going to be functional at a higher temperature. And you also make a good point. Just because it's more functional at a higher temperature does not mean it's more functional at a lower temperature. These sequences evolve in their environment to operate the way that they do in their environment. So when you move them away from their environment, whether that be temperature or pH or some other confounding input, that may change the efficacy of the protein to do the thing that it's supposed to do. To specifically answer your question, the only way to know is to go measure it, really. There isn't really methods right now that you can, with sort of high efficacy, determine whether a protein is going to be active and stable at some specific temperature with a high degree of accuracy. You just have to go test it.
Thank you.
Yeah.
Yeah, I was also curious about this temperature range for practical applications. You mentioned a 30 or 60 degree range and some possible applications. Can you comment on how you would use your results into a practical setup?
Yeah. So, let's assume that we haven't already developed a process for, helping to break down lignin in the output of like a pulp and paper process to, make for us a cleaner waste or something like that. If you haven't done that, which we've already sort of done studies on, but let's assume that you haven't, to simulate some situation where we found some new application that we want to, develop an enzyme. You would first have to find some enzyme that has some degree of efficacy for the thing that you're trying to do. So, for example, there are enzymes out there that have some ability to help break down lignin. You would want to start with one of those, and then you could use the method that I'm suggesting to help you change that protein such that it retains its ability to do that thing at a higher temperature. So right now, the way that you would do that is to run sort of a directed evolution campaign, introduce a lot of random mutations in the sequences, and sort of brute force your way to finding a sequence that can help you break down lignin at a higher temperature with better kinetics or something like that. With this method, hopefully you could get some variation that's better than random and reduce the amount of time that it takes to do that. Does that answer the question?
Yeah, thank you. I don't see any other questions. Thank you for joining us today, and thank you everyone for a wonderful presentation, and we'll see you next time. Thank you.